DNA is a long polymer with deoxyribose and phosphate backbones, having four different nitrogenous bases: adenine, guanine, cytosine, and thymine. One human infection virus that has a DNA-based genome is hepatitis B, which is also a hepatropic DNA virus. Glycoprotein is any class of proteins that have carbohydrate groups attached to the polypeptide chain, also called glycopeptide. They are found on the surface of the lipid bilayer of cell membranes. Some functions would be reproduction, immune system, hormones, and protection of cells and organisms. Coronavirus have a simple protein composition. While there is some variation among different members, a basic set of four protein species universally occurs. The nucleocapsid protein, the spike protein, a small membrane protein, and the membrane glycoprotein. Some coronaviruses have an additional membrane glycoprotein. There are two important regions of a lipid that provide the structure of the lipid bilayer. Each lipid molecule contains a hydrophilic region, also called a polar head region, and a hydrophobic or nonpolar tail region. The phospholipid molecule's polar head group contains a phosphate group. Soap could chemically destroy a coronavirus virion. As a matter of fact, there was an example done on this on social media where a lady had her kids wash their hands as an experiment, and she put, I believe, pepper in the water, and when they put a finger down, all the pepper went away, which proves that soap would be a good way of destroying the coronavirus. Very effective for protecting people. Herpes is a large double-stranded DNA virus that forms latent infections in neural cells of the human peripheral nervous system. Previous work determined that herpes simplex virus genome is found in an ordered nucleosomal structure during latent infection. Studies demonstrate that the optimized binding site and polybasic cleavage site of SARS is the product of accumulated mutations in the receptor binding domain, most likely from natural selection on humans. How might RNA-based genomes result in an infection rate? Viruses exhibit increased mutation frequencies relative to their other organisms. Recent work has attempted to exploit this unique feature by increasing the viral mutation frequency beyond an extinction threshold, and antiviral strategy known as lethal mutagenesis. The central dogma of Molecular biology describes the two-step process, transcription and translation, which the information in genes flow into proteins. Antibiotics wouldn't be effective in treating COVID-19 because they only treat infections caused by bacteria. Antiviral drugs do not deactivate or destroy the microbe, but act by inhibiting replication. In this way, they prevent the viral load from increasing to a point where it could cause pathogenesis, allowing the body's initiate immune mechanisms to neutralize the virus. Most people who become ill with COVID-19 will be able to recover at home. Their bodies will fight the coronavirus and help them get well. To treat other viral infections, they are also being tested to see if they can. An antiviral drug must be able to target the specific part of a virus life cycle.